Muy buenas tardes. Estamos bueno, en esta sesión de la tarde. Vamos a ver esta sesión plenaria a cargo del doctor Jason Hong. Mi nombre es Alberto Goche, soy del INTA Bellavista. Voy a estar a cargo de la moderación y de recibir las preguntas. Jason Hong estudió microbiología en el año 2003 en la Universidad Estatal de Ohio. Es máster en patología vegetal y también en doctorado en plant pathology en la Universidad de Florida. Graduado en máster en 2005 y el PhD, el doctorado en 2011. Su experiencia laboral, bueno, es ecólogo agrícola del USDA de Fort Pierce desde el año 2015 y trabajó en investigación en cítricos y en otros cultivos subtropicales. Es investigador asociado postdoctoral desde el USDA, ingresado ahí, y su unidad de investigación es patología vegetal subtropical en Fort Pierce, Florida. Muy bien, Jason, un gusto tenerte y esperamos tu presentación.
Gracias, Alberto. Gracias por la introducción. Hola a todos. Qué lástima que no puedo ir allá a Argentina. Me encantaría conocer su país y tomar un mate con ustedes y comer un alfajor. Pero ya, esta corona. Espero que me escuchen bien y voy a empezar mi presentación. A ver. Ok. Lo siento, y voy a dar mi presentación en inglés porque yo no sé, bueno, todo el tema, todas las palabras científicas muy bien en español. So today I will be speaking about anaerobic soil disinfestation. It's a technique to control soil pathogens and pests. Just to review, there's a lot of pathogens and pests that limit plant yield and growth. You have the weeds, parasitic worms called nematodes. You have viruses, fungi, and oomycetes. When I was at University of Florida studying with Alberto, I studied work on bacterial wilt caused by Rostonia silencearum. And as you can see, it can devastate a whole field. And this is one of the experiments that we did using thymol to control the bacterial pathogen. Typically, these pathogens and pests are controlled with chemical fumigants. And one of the main fumigants was methyl bromide. It worked very well from the 1950s onward. However, in the 1980s, it was discovered that it was one of the largest contributors to causing the ozone hole. And there was a worldwide ban on creating more methyl bromide and the use of it. And it was one of the first times that all the countries of the world got together collectively to control a substance. Recent research shows that the ozone hole has stopped growing. It's now stable and it's starting to decrease a little bit. However, this left growers with a large problem because they relied heavily on methyl bromide to control these soil-borne pests and pathogens. So currently, they're relying on technology that is almost 100 years old. A lot of them use chlorpicrin, and they use that with other types of chemicals. As you can see, methyl bromide was the optimal. It worked the best for controlling insects, weeds, nematodes, fungi. It was able to disperse well in the soil. The newer chemicals that have been developed since the ban of methyl bromide have not worked as effectively, and some of them are found to be toxic to the environment and to humans, and so they've been banned. There are non-fumigants that are being used to control these soil pathogens and pests. Some of them are biologicals, which are other microbes that are antagonistic to the soil pathogens. However, what they're finding is that sometimes these biologicals do not transfer from field to field, and they can sometimes be very specific up to strain specificity. Other methods are using cover crops which in between planting tomatoes or cucurbits, they'll plant a non-host for these pathogens, which will limit the population. And then a lot of times they'll mulch in the cover crop, which will add a soil amendment, which can encourage these pathogens, or sorry, these a greater diversity of bacteria, which will limit the soil pathogens. Other methods are using heat or steam. This is a steam generator, usually used for high yield crops that you can get a lot of money, such as cut flowers. But again, it's very costly to send steam into your soil. Another technique, which is very similar, is called solarization. It uses a clear plastic mulch, and soil temperatures can reach up to 60 degrees Celsius. And it works very effectively for controlling certain pathogens. But working with a lot of growers with that had Rostonia or bacterial in their soil here in Florida and the southeastern US, they just sell their farms or their land because they cannot control the pathogen. Anaerobic soil desinfestation utilizes these three techniques. It increases biologicals that can control the soil pathogens. A lot of times it uses cover crops or a soil amendment, and it uses a film to protect the soil and also increase the temperature. So I will talk about soil disinfestation. It was developed as an alternative to methyl bromide and other soil chemicals independently in the Netherlands and in Japan. During World War II, the Germans took over parts of the Netherlands and they flooded the farmland that they think was being used by the Allies or as an airstrip. And a lot of that farmland, they let the water out, and then the growers replanted tulips. And what they found was that nematodes and other soil-borne pathogens were controlled for a few years. Thus, the Dutch will use this technique where they will flood their fields for 2.5 to 2 months prior to planting. In Japan, they were using rice paddies as, and they're rotating the rice, and they're putting other vegetables and crops in there. And utilizing solarization and adding amendments, they found that it activated the microbes and they had a higher accumulation of volatile compounds. ASD has also been called biological soil disinfestation, ductive soil disinfestation, and redox soil disinfestation. The term ASD is now is more popular because it emphasizes the lack of oxygen and the growth of anaerobic microbes. It's applied in 3 easy steps. The first step being incorporating organic matter. In Japan, a lot of times they use ethanol. In California, they've been using rice and wheat bran. Other parts of the world have been using soy flour. Cover crops have been used. In Florida, we've used molasses and venesis. Then you irrigate the soil to saturation and cover with a plastic film or mulch. It has been tested with various types of crops, including onions, asparagus, solanaceous crops, strawberries, your cucurbits. It's also applied for trees such as pecans, almonds, and apples. And it's been shown to control fungi, soil-borne fungi, oomycetes, bacteria, and root knot nematodes, another type of nematodes, and also weeds. In Florida, our standard practice is using composted broiler litter, molasses, and then that is tilled into the field. We create this false bed for the amendments to be applied. Then we cover it and we add drip tape below or drip lines underneath the plastic and saturate the soil to fill capacity. So this all came about by looking at the different components and seeing which ones worked best. This was comparing the control in weeds. And what they found was when they had no water, did not control very well, except for using solarization and chicken manure. When they added 5 centimeters of water, the combination of solarization, water, and molasses at 5 centimeters of water or 10 centimeters of water offered the same or similar control as methyl bromide. Looking at fusarium, when they looked at the different amounts of water and with and without molasses, the molasses offered a better control of fusarium than the methyl bromide standard. Then they also looked at root knot nematode, and using saturation and the combination of just solarization of molasses, the chicken manure, or the combination of the 3 of them, it worked better than methyl bromide. We worked with an ag economist to see how economical using ASD was compared to the grower standard or the chemical solarization fumigation. We worked in Citra, Florida, and compared tomatoes. What we found using a half rate or ASD 0.5, so half the amount of molasses, we had a net return greater than the grower standard. Part of that was due to the harvest. The tomatoes plants, we had more tomatoes than the grower standard and a higher marketable yield. So ASD, how we apply it is we apply the molasses, the composted chicken manure, we tarp it with plastic, we add water, and then we let it sit for 3 weeks. Then after 3 weeks, we poke holes in the plastic, transplant, and then we wait till harvest. When I first joined the lab, they wanted to see if there was a difference between the untreated control and the ASD treated, how it affected the bacteria. So we took soil samples prior to transplant. And what we found was that the Bacteroides and the firmicutes populations were higher than the untreated controls. There was a large difference between the populations. However, I wanted to see what happened during ASD, because a lot of times we don't have a full view of everything that happens. So I wanted to do temporal sampling. My boss said it was too much money to do high throughput sequencing, so I had to come up with other techniques to look at the composite of the population. We settled on length heterogeneity PCR, or LHPCR, because of the instruments that we had at the lab. It is good for describing the community. However, it does not say who is in there. So I like this analogy, or this kind of symbol where we have high throughput sequencing. It gives you very good details of what's happening. However, due to cost of money or computational costs, we can't sample as much. So we sample at the beginning of the play or at the end of the play, so we can see this, the goal was scored, but hopefully this works. We don't see all the action of what actually happened during the play. And we have a whole story when we have we can see the whole community. So, So what I did was I sampled during the ASD treatment, that 3-week treatment, and we used LHPCR. And we took, as soon as we took a soil sample, we would cover it with tape to limit how much oxygen got into the system. And we found that the dynamic was very dynamic, the population. There was a lot of microbial shifts going on. But I call this the tale of two fields. Field 1, the microbial population was very dynamic, while in field 2, it was static. as we were taking the soil samples, we also took the soil pH, and we found that the pH, the soil became more acidic, usually within day 1 to day 5, sometimes a little faster, but then slowly throughout the ASD process, it goes back to pre-treatment pH. However, in field 2, there was no drop. The difference between the fields was that in field 2, we had a thunderstorm, a rainstorm. So we applied the manure and the molasses, and then we could not cover it with the plastic tarp. And so quite possibly all the amendments washed away, but we never saw the effect of the ASD effect there. So then I compared the bacteria populations of ASD treated soil and the non-treated soil, and the non-treated soil was taken next to the rows. And so in the open field, in the open air, and it all grouped with the pre-treatment sample. And but you can see it was very dynamic because it was not covered with the plastic mulch. So rain events, cooling temperature events, it was subjected to all of that. While there was big shifts under the plastic in the ASD treated soil, the populations were significantly different from each other. Also taking the pH, the untreated control, the pH had a small drop, most likely that was due to a rain event, but it remained constant. The pH remained, well, it increased a little bit, but it remained pretty constant. But with the ASD treated soil, we saw a significant drop, and then slowly returned back to pre-treatment samples. So part of the questions is how can we get grower adoption? So we have this technique that works very well. So we devised various field and farm experiments where we invited growers throughout the state of Florida and the southeastern US to look at our field plots. So we planted different types of crops. We had a blind test, and they would go and rate weeds, insect damage, diseases, the plant vigor. And so they rate all these things. And then we would talk to the growers and get their suggestions and reveal to them what the treatments were. The growers suggested to not use the solarization plastic because for them, they would have to rip the plastic back up because in Florida, it becomes too hot. They prefer using an opaque plastic. They also suggest using pelletized composted broiler litter, which has been pasteurized because they were concerned about human pathogens such as Salmonella or E. coli. They also were interested in other carbon amendments that would be cheaper than molasses. Molasses is pretty cheap in Florida, but they were interested to see if there was other soil amendments that could be used. We came up with the idea of using algae. So in Florida, we have a lot of cattle ranches, and orange and agriculture is very large. We have a lot of canals that lead the water to stop the swamps and to drain the swamps out. And what happens is you have a lot of this ag runoff, and it has created problems in the oceans. We have algal blooms and fish die. It becomes very toxic. So they've created these algal turf scrubbers, which will filter the water, will send the runoff water, and it will then run through. It's about the size of 2 football fields, and it allows the algae to grow in there. And they harvest the algae, they take it and they just throw it in the dump in the garbage because they don't have anything more to do with it. But what we did was we dried the algae out, then we mulched it and added it as a soil amendment. At the same time we were doing this experiment, there was a paper that was published that talked about when it's in cooler temperatures, you want to use higher rates of carbon because it may offer better control. So in this study, we used high throughput sequencing to look at the different bacterial populations. And we use, here's our control. This is algae, our ASD, and then 2 times the amount of molasses. And we wanted to see how that affect the bacterial populations. And this is post-treatment. So this is 3 weeks after applying the soil amendments. And for all 3 of them, the firmicutes is very high. However, depending on the carbon source used, it depended on the ratios of the bacteria. Using NMR, which is looking at the different spins of the atoms and in the soil samples, we wanted to see what metabolites were being produced and how that related to controlling the pathogens. So what antimicrobial metabolites. And what we found was that depending on the carbon source also, so this is algae, ASD, and 2 times the molasses, we would have different amounts and types of metabolites produced. So we conducted a study where we're like, what if we threw all the molasses in? And we wanted to see what would happen if we had 0 molasses, our standard amount, 2 times the amount, 3 times the amount, and 4 times the amount of molasses in the soil. What we found was that pH of the soil significantly dropped depending on the, and it was correlated to the rate of the molasses that was used. The higher the amount of molasses, the lower the pH, and it never recovered after 21 days. So then we looked at the different organic acids that were being produced. And this is formic acid, which is found in molasses. And so you see a high percentage of the formic acid, and then it significantly reduces as it's being metabolized. What is interesting is that you have valeric acid, and you have certain organic acids that peak throughout the time sampling. So citric acid was being created 1 day after the post-treatment. And again, it correlated to the amount of molasses that was put in there, or the amount of the carbon source. So we're increasing certain populations of bacteria that then create these organic acids. And what is very interesting is that it correlated to the amount of molasses that was added to the soil. This is lactic acid, and it maintained throughout. And again, showing that the more carbon source produced, the greater amount of the different acids that were being created. Acetic acid, propionic acid, isobutyric acid, which was more predominant after or day 21. So then I wanted to see how this affected the biodiversity of the treatment. Of the bacteria in the soil, found in the soil. This is a little busy, but this from here to here is soil samples that were taken from 0 to 15 degrees or 15 centimeters. So the top portion of the soil to the lower bottom portion of the soil. And this is treatment 1, which had 0 molasses. And the biodiversity changed, but it was not as drastic compared to, this is treatment 2, which is our standard rate. The biodiversity dropped, and then it slowly increased during the time period. Looking at 2 times the amount of molasses added, the biodiversity again dropped more significantly than the standard rate, and it slowly increased. And we saw the same pattern with the lower population, but with the 4 times amount of molasses, it reached the lowest diversity index, meaning that it lost the most diversity, and it was not able to recover to pre-treatment samples. So this is pretty busy, but what we're showing here is that the no molasses grouped with the day 1 based on bacterial populations, and they had the most diversity, while you have 2 times the amount, our standard rate, and the 4 times the amount of molasses, reduce the population, and these numbers represent the time sampling, the days that we sampled post-treatment, and the actinobacteria, the phylum, reduced, but we had an increase of firmicutes and proteobacteria, which are known to be anaerobic. This is the bottom from 15 centimeters down to 30 centimeters. But what does this all mean? We're just showing that, who's being affected by this. Well, when we planted the tomatoes, we added 0 molasses, our standard rate, 2 times, 3 times, and 4 times. The more molasses that was used, it was too acidic for the tomato plants. And we had a 10% mortality rate to a 35% mortality rate. Rating the plants on a scale of 1 to 5, we found that you should not add too much molasses to your soil. The pH did not recover. However, it was very effective for controlling soil pathogens. As many of you know, HLB, or citrus greening, is problematic here in Florida. And we wanted to see what would happen if we planted citrus trees in ASD treated soil. So we used high throughput sequencing and comparing the untreated, which is the blue, and the treated soil post-treatment. What we found was that ASD reduced the bacterial diversity, increased the proteobacteria compared to the untreated control, and Bacteroides was a lot higher and Firmicutes was a little bit lower. But what we found when we went back to these trees 2 years after applying ASD, that there was still an effect where we sampled the soil near the root zone and extracted the DNA, used high throughput sequencing. We found the trees that had received no ASD, so the untreated trees, they were not different, they grouped pretty similar. And those that received ASD, those grouped separately. And there seemed to be an interaction with the rootstock. We used sour orange and swiggle. And using a pairwise test, we found that swiggle was significantly different from ASD to untreated control, while sour orange also was different with the bacterial populations. ASD treated soil, there was greater actinobacteria, and then firmicutes. While you look at the untreated control, the firmicutes and the acetobacteria were greater there. So it was flipped for those two, for the second and third most populous phylum. But what does this mean? How does this affect the crop? Using drone imagery, we looked at the ASD treated trees and the non-treated trees, and we found that the ASD treated trees had a greater canopy than the non-treated trees. This was taken 4 years after planting, after the ASD treatment. Unfortunately, all the trees tested positive for HLB. However, what we found that they were a greater height, greater fruit yield, greater weight, which was significantly different, and the stem diameter. Remember, these are grafted. The stem diameter was greater for the ASD treated trees compared to the non-treated trees. So what we're currently doing in the lab is we're looking at making a byproduct or a product that could be applied. I'm interested in creating almost like a probiotic for the soil, where we add the activated bacteria and we add it to the plants. This, because right now it requires 3 passes with the tractor to create for ASD. My hope, what I'm trying to do is create a product where we just apply it once and we can apply it with either through the drip lines or through a water truck. My main concern was, would the byproduct, or would the product that we created, would it cause the same mortality rate in tomatoes that we saw with the added molasses? And fortunately, it did not. We observed that it did not have the same anaerobicity power as the standard ASD. However, we used field soil and it controlled weeds just as well as ASD did. We're also looking at what kind of gases are being produced. One of the concerns is the amount of greenhouse gases that are being produced with ASD. And we're trying to work to see if we can mitigate that or how much is actually being produced compared to using chemical fumigants. Another area of interest is working with blueberries. Blueberries has become a big crop in Florida. However, one of the soil-borne pathogens called Rostonius lanciarum is infecting blueberries. And so one of the areas of research that we're looking at is using ASD as a replant to disinfest whatever may have caused the death of these plants. And so we would take a small square, apply the ASD here, wait 3 weeks, and then replant the bush or the tree. We're also looking at biodegradable mulch and comparing that to the plastic to limit how much plastic we're using, and looking at different types of cover crops and comparing that to molasses. And that concludes my talk for today. I'd like to thank all the collaborators, the postdocs, and technicians, the various people that have entered and helped out with this research. And I'd like to entertain any questions.
Ok, muy bien, Jason. Verdaderamente muy interesante. Con respecto a lo de los productos bioestimulantes que están desarrollando, o sea, la idea es producir un producto químico que favorezca la proliferación de las bacterias o llevar las bacterias, las mejores bacterias al suelo y ponerlas ahí.
Bueno, yo voy a contestar en inglés.
Está bien, está bien.
One of my thoughts is that looking at this as an ecologist, there's certain bacterial populations that are found at each field, and they require different amendments, or there's different nutrients that are in there. What I would like to see is we use the field that's found in the grower, the bacterial populations that are found naturally in that grower's field, and increase the bacterial populations or the antimicrobial populations of these fungi or whatever it may be, to increase that naturally in the soil and to amend those and for that to grow. So it could be a amendment, like you said, or we could take their soil and increase those populations there by utilizing this technique of putting it in flasks or putting it in vats and creating the byproduct that way.
Bien, la pregunta venía porque aquí está habiendo muchos productos que son el líquido que sobra de cuando hacen compost, de, por ejemplo, manure chicken, esas cosas de gallina,
Sí.
Y sale un líquido, y ese líquido lo mezclan y tiene muchas bacterias, y eso lo usan para agregar al suelo, y resulta. Y ahora la gente sabe que es muy fácil hacer eso, y ahora hay muchos de esos productos, y no están llevando un químico, están llevando la bacteria. Ahora bien, una de las preguntas del público es si, bueno, no sé si por ahí se nos escapó, esos ensayos en donde se veía el control de las enfermedades, ¿se agregaba la enfermedad en alta población? ¿Eran suelos naturalmente infectados o ustedes ponían la enfermedad en el suelo?
En respecto de este manure, ese líquido que sale del manure, realmente the pathogens that might be in that manure should not, I don't know if you're actually putting plant pathogens in there. We'd have to test that. However, you are increasing the bacterial population, and typically the rule of thumb was the greater the bacterial diversity, the microbe diversity, it decreases the population. However, we are thinking now, maybe we need to look at more as a function. So what is the function of the bacterial population or the soil microbiome? And is that inhibiting the plant pathogens? Yeah, so we've also looked at, we call it green manure or liquid manure. If we use that, the one concern, and going back to your previous question, is you take bacteria from one field, sometimes it does not live very well in another field because it's missing some of the nutrients. And it'd be the same with this increase of the liquid manure. Will it be able to survive long term in that soil? It's like if we took you took me and dropped me off in Argentina or in the North Pole or the South Pole, with the polar bears. I can't survive there. Or you put me in a desert because I don't have the proper equipment, or the food, water, things like that. So that's where I'm more interested in. Can we amend the soil to bring up these populations that are currently there, that are possibly there, and that will control for these plant pathogens?
¿Tienen alguna experiencia o algo de trabajo con, por ejemplo, pasteurela, alguna de esas controladores biológicos de suelo? Son bacterias que controlan nematodes.
So we've looked at, I believe, the previous research. So we had nematologists that I worked with. We found a lot of the bacterial populations that belong to the phylum Actinomycetes or the acetobacteria seem to control nematodes, and they will amend the soil with chitin because they seem to like chitin. And nematodes, the outer layer is made of chitin, so you're increasing that population, and hopefully that's controlling the nematodes.
Bien, bueno, felicitaciones por el trabajo también. Con respecto a al agregado de búsqueda, o sea, sería la búsqueda de suelos sanos, healthy soils. Lo ideal sería para el tipo de suelo, porque, por ejemplo, Florida tiene un suelo también arenoso, pero es más alcalino, y donde vivimos nosotros tenemos un suelo arenoso más ácido. No, la misma población no se podría repetir porque son ambientes distintos, pero si se podría llegar a hacer una receta de quiénes tendrían que estar en cada tipo de suelo, uno podría hacer esto comparable y llevarlo a cada lugar del mundo. Podría uno tener, extrapolar los resultados de un lugar a otro. ¿Eso sería factible?
Bueno, el consejo que estamos dando es que cada área tiene su propio carbón que se puede utilizar. Entonces, quizás en su área, quizás hay mucho mate que pueden entregar en la tierra que pueden usar. Para nosotros tenemos mucho de esta molasses o sugarcane, entonces es bien barato. Es mejor usar las cosas, materiales baratas para en vez de para que the growers adopt this technique.
Okay. Agregué una pregunta que te la hacen en inglés, por las dudas que no la diga bien, por si la quieres leer. Es, how do you bring bison standardizing this procedure, no sé si la puedes leer, for different type of soil. That's it, standardizing the population of microorganisms.
What has been interesting, there doesn't seem to be a specific recipe or a specific technique for using this. This has been tried throughout the world, far north into the Netherlands, in Canada. They've used ASD. They've also used it in very tropical regions like Florida and Japan. And it seems to work every single time. Sometimes the technique, depending on the temperature, you need to have ASD be longer. In the Netherlands, they recommend doing it throughout the winter time, so for about 6 months. And they're using grass or a different amendment for their amendment. In Washington State, where they grow apples in the United States, they use grass also, and they have it sit for 3 months. So, but what they're finding is the combination of adding the carbon and the water, and then covering it with the plastic mulch, the soil with the plastic mulch, that seems to work best for soil pathogen control.
Okay. Bueno, Jason, no tenemos más tiempo, pero podríamos seguir hablando un buen rato más. Quiero agradecer a vos primeramente por tu tiempo, a la gente que ha preguntado, a los que han participado mirando esto. Esto quedará grabado a futuro en la página del canal de la asociación. Dentro de una semana será cargado. Y los invitamos a todos que visiten los stands de los sponsors comerciales y también los pósters. Y para hoy a la noche, acuérdense que también tenemos el cierre con la entrega de premios. Y quédense conectados, siguen las siguientes presentaciones. Gracias, Jason, y nos vemos la próxima.
Gracias a todos. Y quiero conocerle cara a cara, si posible.
Bueno, ojalá se pueda. Hasta luego.